Yeah, we used a very interesting technique, very exciting technique called induced pluripotent stem cells, where we can take a very small amount of a patient's either skin sample or a blood sample, and we can reprogram this into what's called a pluripotent cell, which is then differentiated, made to be a retinal ganglia or retinal organoid. So recapitulating the normal cells that you have in your eye, and that allows us to model the cellular architecture of age-related macular degeneration using a patient's own cells. I think fundamentally we've made an important stride forward in understanding the way in which the genetic differences between people ultimately act to cause the disease. And for us, the next really important steps are to work on two strategies. One, using gene editing technologies, CRISPR technology, to see whether we're able to correct for the genetic mutations that cause these diseases. And secondly, to use our induced pluripotent stem cell systems to create a platform to screen a large number of drugs and compounds to determine which ones are most likely to have a beneficial effect on patients. We've learned that many diseases really act at the level of individual cells, and so to study the disease we need to be able to use the cells where that disease is occurring, it's pathogenic. And for obvious reasons, accessing cells in your retina or your optic nerve are almost impossible, very challenging, and so we use stem cell systems to be able to recapitulate and recreate these cells, but starting with a patient's own genetic material. So it carries the same genetic differences between different patients and recreates the same cell subtypes that you would normally have in your eye and your optic nerve.